All right, so I guess we begin. Okay, hi everybody. My name is Sabra, Salim.
And I'm Ashley Adile.
And we are here from McMaster University. We are undergraduate students in the laboratory of Dr. Sheila Singh. Cool. So we had our case study. So we had our patient, Jacques, and he presents with the following symptoms. So lethargy, headaches, forgetfulness, and mild left-sided weakness. So after diagnostic and pathological testing, it was determined to be glioblastoma. So what is glioblastoma? Glioblastoma is a highly aggressive brain tumor. It's one of the most common primary malignant brain tumors in adults. And despite surgical resection, as well as a standard of care, so with chemotherapy using the alkylating agent temozolomide and radiation therapy, relapse is almost inevitable. On top of that, the median survival only sits at around 15 months, with usually uniform fatal prognosis. A lot of this can be contributed to the cellular and genomic heterogeneity of the tumor. So when we look at the different subtypes of GBM, we have four. So we have the classical, the mesenchymal, the neural, and the proneural. And although the survival rates seem to be, there aren't really any distinct differences, we can see in the left sample just over here that the transcriptomics of the tumors are very different, so they're very diverse. So what does this mean? This basically just means that the genetic landscape of the GBM tumors are constantly evolving. And this is a spatial and temporal evolution. So the therapy that we want to look at, so we're looking at the cellular heterogeneity of the tumor. So we're looking at this type of brain cancer stem cell known as the brain tumor initiating cell, or BTICs. And through therapy, or through the literature, it's been determined that these types of, or this subpopulation of cells is able to evade chemotherapy and radiotherapy. And they also have the stem cell property, so namely self-renewal and multilineage differentiation. On top of that, they're able to evade this type of chemo and radiotherapy through clonal evolution. So what we hope to do is we hope to target this subpopulation of cells, and in order to do this, we will be looking through one therapy. We'll look at BMI1, which we'll mention later, and this hopefully will be able to eradicate the tumor as a whole and attack the cells that are responsible for tumorigenesis. So we look, hopefully, again, to target BMI1, and hopefully this will prolong patient prognosis. So why have we chosen BMI1? So BMI1, we know, is responsible for or promotes self-renewal and proliferation in these cells. And in the literature, in comparison to controls, decreased BMI1 expression has increased survival in vitro assays as well as in mice models. So our hypothesis is, using a combinatorial approach in conjunction with standard of care, we hope to target the BMI1 in order to find some sort of innovative treatment for these patients.
Now, how do we do this? We're hoping to propose looking at functional validation of BMI1 inhibitors in vitro assays, taking the most efficacious ones and putting them into our mouse-adapted, patient-derived mouse therapies, looking not only at the drug itself, but also in combination with standard of care, as well as performing a CRISPR screen to identify the GBM therapy-resistant genes. In terms of methodologies, this is just a slew of in vitro assays that we would like to perform, looking at the BMI1 protein levels and targeting them. We would like to start off by saying that in our lab, we are fortunate enough to have collaborations with surgeons that provide us with newly resected GBM samples, as well as other brain cancers, allowing us to propagate cancer stem cell lines, allowing us further to look at in vitro assays. So first, proliferation assay, or IC50 curve, IC50 being the concentration at which, the concentration of the drug at which 50% of the cell's proliferation is inhibited. Next being a mixing experiment where human neural stem cells along with GBM cells are mixed together, and upon administration of the drug, we would expect that normal neural stem cells are spared, having an increase in the normal neural stem cells and decrease in GBM cells present. We would also look at BMI1 protein and transcript levels. Aside from this, with a secondary sphere formation assay, we would look to the spheres, measuring the average amount of spheres in the control, so DMSO control, as well as those treated with the drug. And spheres are indicative or correlate to the number of BTICs. Therefore, an increased number of BTICs would lead to worse or more aggressive prognosis. So we would hope that in the control, we would see more of these spheres and less in the drug-treated ones. Last would be a cell cycle analysis. So normal cells go through cycle through 4 different phases of cell division, and we would expect that those treated with the drug would be geared towards apoptosis or cell death. In terms of our mouse model, we would inject human GBM cells into mice and looking at their tumor progression across this timeline, as well as their tumor burden and hopeful survivor benefit, specifically with the second cohort of standard of care, so temozolomide and radiation, that would target the bulk tumor, and then the BMI1 inhibitor afterwards, we would specifically target the BMI1 cells, and it would be evaluated across these various time points, so ranging from engraftment to recurrence. And taking these, we would combine it with the Cas9 system, and this is proposed to, it's a novel system or technique that knocks out genes. And with this, we would have the sole purpose of looking at GBM therapy-resistant genes as well as those driving GBM from its primary state. So with this, we would identify the treatment refractory genes, looking at the GBM putative drivers, or the genes that drive GBM, looking at them through the assays that we explained in AIM-1, looking at inhibitors afterwards for those such genes, and co-targeting them with the BMI1 inhibitor, similar to the second AIM. So we believe that this is novel work as it combines both standard of care, which current GBM patients receive, as well as other targeted therapies, and it's seen to be more successful than monomodal therapies. Aside from this, it also promotes collaboration with industry partners, and even further to this, it's able to elucidate signaling pathways in brain tumor and brain tumor recurrence. But it does not stop here. Equally important is the support that patients and families receive, and with that, we are very fortunate to be working in Dr. Sheila Singh's lab alongside her, as well as the many innovative scientists that we are grateful for as mentors. So we definitely thank them. And a big thank you to the Brain Tumor Foundation of Canada for giving us this opportunity to be presenting amongst many talented students. Thank you.